All right, we're now on topic 4, which is cell transport. That's what we most recently did in class. Remember, transport is talking about the movement of molecules into and out of the cell. The function of the cell membrane, which is our first bullet point, says that it selects what goes in and out of the cell based on size. Another term for this, selecting what can go in and out of the cell, could be selectively permeable, meaning that it selects, right? Remember, the membrane has tiny little pores and only certain molecules can fit in. So how do we decide which molecules can fit in? Well, if we're basing it on size, small molecules can. What are some examples of small molecules that can get through? That's going to be glucose, water, and then iodine. Remember, iodine was that amber material that we used in our cell lab. The other one that we have are large molecules. Large molecules cannot fit through the cell membrane. Examples of these would be starch. Starch is the one that we want to make sure that we really know, and the reason for that is because that was tested on the state lab. Other ones that can't fit through include proteins, and then also fats. Remember, you can also call fats lipids. The only way that they can get through the cell membrane is that they need to be digested into smaller molecules. Those smaller molecules are going to be their building blocks. So for example, starch, a large molecule, would need to be digested into simple sugars. Remember, an example of a simple sugar is glucose. The final property of the cell membrane is that it can recognize certain chemicals using protein receptors. Remember, proteins are going to have a specific shape, and that specific shape of the receptor is going to link up with, let's say, a hormone or a neurotransmitter, which has been released and then binds to that receptor so that a chemical message can be transmitted. This also is called cell-to-cell communication. Cells in your body are actually capable of talking to one another. What allows them to do that? Again, it's the receptors and then the chemical messengers, the neurotransmitters and the hormones that are released by cells. Next up, we have there are two different types of transport. The first type of transport that we're going to talk about is passive transport. Passive transport also goes by the name diffusion. They're synonyms, so they mean the same exact thing. Diffusion is the movement of molecules from high to low concentration. Remember, this doesn't require any energy. It's like riding your bike downhill. So I always try to remind you, remember, diffusion and downhill, the two D's go together. The easiest way to remember this is to make sure that you go and you draw your little graph. When you're drawing your graph, remember, when we're talking about going downhill, it's like we start at the highest point. So here I have my high. It's like I'm going downhill and then I'm going to my lowest point. When you ride a bike downhill, think, do you have to pedal? No, you don't have to pedal. Therefore, there's no energy, right? No additional ATP that you're going to have to add in order for those molecules to move. So small molecules can easily pass through the cell membrane through this process known as diffusion. The other type of passive transport is going to be osmosis. Osmosis is talking about the diffusion of water. Now, if you're unsure if it's diffusion or osmosis, always write down diffusion. And the reason for that is because osmosis is just a super specific type of diffusion, whereas diffusion is all encompassing. So it actually includes diffusion of water, diffusion of glucose, diffusion of oxygen, diffusion of carbon dioxide. The next one we have is active transport. Active transport is going to be when the molecules go from low to high concentration. This one requires ATP. It's like going uphill. Again, what I really like to do is draw that diagram. This time, it's like I'm starting at the bottom of the hill, the low point. So I'm going from my low concentration up to my high concentration. If I'm biking uphill, I'm probably going to be pedaling really hard. Since I'm pedaling really hard, I know that I'm going to need that ATP to help me get up the hill. Another thing to note about active and passive transport is that you always have to count the molecules. I'm telling you, even if I'm answering a question about active and passive transport, I'm counting those molecules. It's very difficult to answer them if you're simply guessing how many molecules are on either side. So here I have an example, and we've seen this example a couple of times on our tests, on our do now, and on our homeworks. It says, whenever there's a transport question, remember to count the number of molecules on both sides. Says determine if the following scenarios are active or passive transport. When I see active, again, what I want to think is ATP. When I think passive or diffusion, they mean the same thing. I think about going downhill. So I'm starting high, going to low. So it says molecule B moving into the cell. First thing I do is I hey, what does molecule B look like? That's going to be the square. So I go and I count up how many I have on the outside. I have 2 on the outside there. Then I count how many I have on the inside. 1, 2, 3, 4. 4. Here it says moving into. Sometimes people are uncertain. What way do I draw the arrow? It's always going to tell you in the question. I go and I draw my arrow that way. So now I'm talking about going from 2 to 4. Your best bet, make sure that you identify which one is the high number and which one is the low number. The high number in this case is going to be 4. The low number is 2. So we're going L, right, low to high. If you need to, sketch that out. That means we're starting at the low point of a hill. So I start here at the low, and I'm going up to that high point. If I'm going uphill, do I need energy? Yeah, right? It takes a lot of energy to get uphill. So that's going to be ATP. In this case, that means that this type of transport is going to be my active transport. My next example, I do the same thing. Let's use a different color so it's a little bit easier for you to follow along. Molecule C. When I look at molecule C, I realize, hey, I'm looking at the triangle. So now I have a new thing to count. When I look at the outside, there are actually none on the outside, so that's 0. Then I count how many are going to be on the inside. There are 7. Once again, note which way the arrow is going to go. In this case, it says out. That means those molecules are going to move out of my cell. So I have 7 to 0. Again, I identify which one's the high number. My high number is 7, and my low number is 0. I draw that little graph to help me. I'm starting off at the high point, going to the low point. If I'm going downhill, remember, downhill is my hint, I'm talking about diffusion. And diffusion is really just another word for passive transport. So again, the key, whenever you see a question like this, you want to make sure that you count up those molecules so you can figure out if it's active or passive transport. The next section is looking at the state lab. Now remember, when we were doing the state lab, we made a little tiny model cell, and then we needed to see what was going to happen inside of that model cell. This is what the setup looked like. It was called diffusion through a membrane. And it wants to know, what is the after going to look like? Remember, we waited about 10, 15 minutes and we saw where those molecules moved. The first thing I always like to do is I look over here at this key. And I'm going to identify what molecules are considered big and then what molecules are considered small. So I have starch. Starch is big. Remember, it's made up of a lot of simple sugars all bonded together. Then I have glucose. Glucose is going to be small. It's just one simple sugar. And then starch indicator, which also goes by the name iodine, is also going to be small. Anything that's small, it can easily diffuse. It's not going to need to be digested. Because of that, iodine initially is on the outside. Iodine still stays on the outside, but it's capable of diffusing in. Remember, when it's equal on both sides, we call that equilibrium. Next up, we have glucose. Glucose, also a small molecule. That means it's going to be on both sides because it's able to diffuse out of the cell. And then finally, starch. Starch is a big molecule. It's not going anywhere. It's stuck inside of the cell. Just kind of as a little bit of a reminder, remember our initial color here on the before was amber, and then the inside, it was kind of like a milky white color. If we're looking on the right-hand side, our end result was that this cell, right, that's what this says. What color did the cell turn? It turned a blue-black color. And then it says, why did this happen? This is because iodine diffused in and reacted with the starch. Remember, a chemical reaction occurred here. And that chemical reaction between iodine and starch is what caused that blue-black color. Now, the last section is actually dealing with osmosis. Osmosis was when we looked at the red onion cell underneath the microscope. So osmosis is the movement of water from high to low concentration. Remember, this is really just a fancy type of diffusion. If I'm looking here at these two images, I have on the left-hand side my original onion cell, and on the right-hand side, my onion cell after I added a solution. What type of solution did I add to this? The type of solution that I added to this was salt water. Remember, salt water lowers the water concentration. That means that there's a lot less water outside of the cell. Because of that, there's high water concentration on the inside, low water concentration on the outside. Water is moving out of the cell from high to low. So we have water moves out of cell from high to low concentration. Another acceptable answer would just be like water moves out because of diffusion. Water moves out due to osmosis. And then the final thing we have are the indicators. First indicator is Benedict's solution. Make sure you study this. I know a lot of people struggled with this on the test. Benedict's, when it gets mixed with glucose and it's heated, that's the key. Remember, when they're heated, it's going to turn to a orange color. And Benedict's initially was that light, bright blue color. That was the initial color. And then iodine, remember, was initially that amber color. And when it's mixed with starch, that's going to turn blue-black. And again, I would make sure I look over this because I know a lot of people struggled with this on the exam.